This method can help answer key questions in the cognitive neuroscience field.
The main advantage of this technique is that it can analyze neural activity and connectivity at high spatial and temporal resolutions.
To begin analysis of intracranial EEG data, set up SPM12 and select the MEG EEG analytical menu. First, perform time-frequency analysis for the pre-processed intracranial EEG data of each trial using continuous wavelet decomposition with Morlet wavelets based on predefined parameters. To reveal the temporal evolution of spectral components, conduct wavelet decomposition using 7-cycle Morlet wavelets for the entire epoch of 1000 to 2000 milliseconds, using the frequency range of 4 to 300 Hz. Next, determine the mother wavelet and the number of cycles, and note that the number of cycles controls the time-frequency resolutions and should be greater than 5 to ensure estimation stability. Determine time and frequency ranges, then crop the resultant time-frequency maps automatically to remove edge effects. Here, the maps are cropped at 200 to 500 milliseconds. Perform data transformation if desired, and baseline correction by selecting time-frequency rescale for the time-frequency maps to better visualize event-related power changes and improve normality. Next, convert the time-frequency maps into two-dimensional images. Also smooth using a Gaussian kernel with a predefined full-width